The culturing and analysis of milk from the bulk tank can be a useful tool in maintaining or achieving good milk quality. Knowledge of how samples are analyzed at the lab can help you apply your results on the farm. In this video, several common bulk tank cultures will be demonstrated, and the advantages and limitations of bulk milk culturing will be reviewed. Because there are no official industry standards for bulk milk culturing, each lab may run cultures in a slightly different way, but the general principles and interpretation for each culture test is the same across labs. The tests shown in this video will include the standard plate count, coliform count, preliminary incubation count, laboratory pasteurization count, and Staph aureus and Strep Agalactia detection. Bacterial culturing in the lab begins with selecting a food or medium for the bacteria to grow on. The media is stored in either a petri dish or plate, or in a petri film. For the standard plate count, a media that will grow almost any kind of aerobic bacteria found in milk is selected, typically a media called plate count agar. Plate count agar is also used for the preliminary incubation and laboratory pasteurization counts. For coliform bacteria, MacConkey agar is typically used, which supports the growth of gram-negative bacteria and prevents gram-positives from growing. Coliforms will appear bright purplish pink. To detect Staph aureus and Strep Ag, special kinds of media are used that contain antibiotics to prevent the growth of other kinds of bacteria, as well as supplements to encourage the growth of these pathogens. In our lab, we used Baird-Parker agar to detect Staph aureus, and Edwards modified agar with colistin sulfate and oxalanic acid, or EMCO for short, to detect Strep Ag. Colonies that grow in these agars that look like they might be one of these contagious pathogens are isolated and identified by more rigorous biochemical tests. Once the media is selected, a predetermined amount of milk is added to the plate and spread out evenly to ensure there is a uniform layer. The plates and/or films are then incubated at 32 degrees Celsius for 2 days. After the plates have grown, they are taken out and the bacterial colonies are counted. The colonies represent the bacteria count in the original milk sample and are reported as colony-forming units, or CFUs, per milliliter of milk. For the lab pasteurization count, the sample is pasteurized in a 63.9 degrees Celsius hot water bath for 30 minutes. The milk is then plated onto the agar and incubated. A preliminary incubation count is set in a 12 degrees Celsius refrigerator for 18 hours and then plated and incubated. This encourages the growth of bacteria that like to grow in cooler temperatures. The best rule of thumb for interpreting bacteria counts in milk is the lower the better, but here are some general benchmarks for bulk milk samples. A good standard plate count would have a colony count of less than 5,000 colony-forming units per mil and shouldn't exceed 10,000. Preliminary incubation counts will almost always be a little higher than a standard plate count from the same sample, but shouldn't exceed 50,000 colony-forming units. You can see what these counts look like on the actual plates. The milk on the left is much cleaner and has a bacteria count of around 1,000 colony-forming units per milliliter. The milk on the far right is much more contaminated with bacteria. This plate has over 10,000 colonies on it. These are MacConkey plates. The plate on the far left is ideal and has no coliforms. You can see a few coliforms on this plate, and way too many on the plate at the far right. This preliminary incubation count grew so many bacteria from the original milk sample after it sat in the 12 degrees Celsius fridge that you couldn't even tell the colonies apart when it was plated and grown. In this case, dilution should be used to separate the bacteria out and make them more countable. Bulk tank cultures are not a replacement for individual cow sampling, but they are a useful tool. Bulk tanks can tell you if there is a contagious pathogen present, such as Staph aureus or Strep Ag. Bulk tank cultures can also give you an idea of what types of bacteria are predominant and their approximate numbers. Bulk milk culturing could detect if you're having a coliform problem or not, or if there are a tremendous amount of cold-loving bacteria in the milk. Bulk tank culturing can't help you with specific cow mastitis questions. It is not a replacement for individual cow sampling. Cultures are also not able to tell you where the bacteria are coming from. There is research that associates different types of bacteria with different kinds of contamination, but in order to know where the bacteria are coming from in each specific case, Changes must be made on the farm and new bulk tank cultures run to see if the numbers have come down. Cultures can also not confirm the negative status of a herd for a pathogen. The pathogen might be in such low numbers that it will rarely be detected with a small sampling size. Running multiple samples over a period of time will add more credibility to a negative diagnosis for a farm. Great care should be taken when trying to interpret data from only one bulk tank sample. Culturing the bulk tank 4 or 5 times in a row at different milkings or on different weeks is recommended before trying to interpret culture results.